There is a man whose right arm was worth more to global medicine than almost anything else in his body. Not his brain, not his heart, his arm, or more precisely, what ran through the veins in that arm. For 64 years, he rolled up his sleeve every 2 weeks and let a needle draw plasma out of him. And doctors on the other end of that plasma turned it into a medicine that is now credited with saving the lives of more than 2 million babies. He wasn't a scientist. He wasn't a doctor. He never took a single dose of anything experimental. He simply had blood that almost no one else on Earth had, and when researchers finally sequenced and understood exactly why, the answer traced back through his own childhood, through a branch of human genetics most people have never heard of, and through a quirk of immunology so specific that Australia, even today, only has a couple hundred people who can replicate it. His name was James Harrison, and this is the story DNA tells about him. Harrison was born in December 1936 in a small town in New South Wales, Australia. At 14 years old, he underwent major chest surgery, a procedure serious enough that one of his lungs had to be removed. To survive the operation, he needed transfusions, more than a dozen units of blood from strangers he would never meet. He spent months recovering in the hospital, and by his own account, it was during that recovery that his father told him something that stuck with him for the rest of his life. That he was only alive because other people had been willing to give away part of themselves. Harrison decided, right there as a teenager, that as soon as he was old enough, he would start giving blood too. He turned 18 in 1954, and he began donating almost immediately. What none of them knew yet, not Harrison, not the doctors, not anyone, was that those very transfusions he received as a 14-year-old had done something unusual to his immune system. They had, in effect, planted the seed of the discovery that would define the rest of his life. To understand why, you have to understand a piece of genetics that most people only encounter once, usually on a form at a blood bank. The Rh factor. Blood type isn't just A, B, or O. Layered on top of that system is a second one, the rhesus system, named after the rhesus macaque monkeys, in which it was first identified in the 1940s. The most clinically important antigen in that system is called the D antigen, and whether or not your red blood cells carry it is controlled by a gene called RhD. If you inherit a working copy of RhD, your red blood cells display the D antigen on their surface, and you are what doctors call Rh positive. If you inherit 2 non-functional or missing copies of that gene, your cells don't display the antigen, and you are Rh negative. In most white populations, roughly 15% of people are Rh negative, largely because a substantial share of them carry a complete deletion of the RhD gene rather than just a broken copy of it. In many African and East Asian populations, the Rh negative frequency is dramatically lower, often in the low single digits, and the underlying genetic mechanism is frequently different too. Researchers have found that a meaningful share of Rh negative status in some African populations comes not from a full gene deletion, but from a hybrid RhD-CED gene structure that still results in a negative phenotype through an entirely separate genetic route. In other words, Rh negative isn't one single genetic story worldwide. It is several different genetic stories that all land on the same clinical outcome, which is itself a small window into how much hidden diversity sits underneath categories doctors treat as simple. Here's why that distinction matters for pregnancy specifically. If a woman is Rh negative, and she is carrying a baby who inherited the Rh positive gene from the father, there is a risk that during pregnancy, and especially during delivery, a small amount of the baby's blood can cross into the mother's bloodstream. Her immune system, which has never encountered the D antigen before, can recognize those fetal red blood cells as foreign material and begin manufacturing antibodies against them, specifically an antibody called anti-D. In a first pregnancy, this usually isn't dangerous, because sensitization typically happens too late to affect that particular baby. But once a woman's immune system has been sensitized and has anti-D antibodies circulating in her blood, every subsequent pregnancy with an Rh-positive baby becomes progressively more dangerous. Those maternal antibodies are small enough to cross the placenta, and once they do, they attack the fetus's red blood cells the same way they would attack an infection, breaking them down faster than the fetus can replace them. The result is a condition called hemolytic disease of the fetus and newborn. In its mild form, it causes anemia and jaundice. In its severe form, it causes massive fetal anemia, heart failure, a condition called hydrops fetalis, and death, either before or shortly after birth. Before medicine had any way to intervene, this condition killed or permanently disabled tens of thousands of babies worldwide every year, and it was one of the most feared complications in prenatal care for the entire first half of the 20th century. The breakthrough that changed this came in the 1960s, when researchers in the United States, the United Kingdom, and Australia, working somewhat in parallel, figured out something that sounds almost paradoxical. If you inject an Rh-negative mother with a carefully measured dose of anti-D antibody right after she delivers an Rh-positive baby, or after any event where fetal blood might have entered her circulation, you can stop her own immune system from ever mounting its own sensitized response in the first place. The donor antibody essentially finds and clears the small number of fetal Rh-positive cells from the mother's bloodstream before her own immune system gets a good enough look at them to build a lasting memory response. It's a form of immune deception, borrowing someone else's antibody to prevent your own body from ever learning to make it. Researchers are still refining the exact mechanism today. Some studies point to rapid antibody-mediated clearance of the fetal cells by the mother's spleen and liver before an immune response can form. Others point to more subtle immune regulatory effects layered on top of that clearance. But the clinical result has never been in question. Once the shot, known as Rh immunoglobulin or anti-D immunoglobulin, became standard practice for Rh-negative mothers in the late 1960s and 1970s, cases of hemolytic disease of the fetus and newborn collapsed by something close to 90% across the countries that adopted it. There was just one enormous catch. To manufacture that shot, you need a raw ingredient, high-titer anti-D antibody, harvested from the plasma of a small number of Rh-negative people whose immune systems produce large amounts of it. And you can't simply ask any Rh-negative volunteer to show up and donate. The antibody has to already be present in their blood at a meaningful concentration, which almost always means the donor was, at some point, exposed to Rh-positive blood cells, either through a transfusion or, in the case of women, through a pregnancy with an Rh-positive baby. Most Ra-negative men have never been exposed to Rh-positive blood at all, so their plasma contains none of the antibody the manufacturers need. The pool of people who are simultaneously Rh-negative, who have been sensitized, and who produce a strong, stable, high-titer antibody response is small everywhere in the world. In Australia, at any given time, the blood service has relied on a donor pool numbering only in the low hundreds to supply anti-D immunoglobulin for tens of thousands of mothers and babies a year. James Harrison turned out to be one of those people, and an unusually potent one. When he began donating blood in 1954, doctors soon discovered that his blood carried an extraordinarily strong and consistent anti-D antibody response, almost certainly the product of the transfusions he had received as a 14-year-old during his lung surgery, which had exposed his immune system to Rh-positive blood at a formative moment and left behind a durable antibody memory. Once Australian researchers understood what they had, in the mid-1960s, right around the same period the anti-D treatment itself was being developed and trialed, they asked Harrison to switch from donating whole blood to donating plasma specifically, since plasma could be collected more frequently and in a way that let his body regenerate the antibody-rich fluid faster than red blood cells could regenerate. He agreed, and from that point on he donated plasma roughly every 2 weeks, without missing an appointment, for the next 50-plus years, despite having a lifelong, genuine fear of needles that he talked about openly. He would look away every single time. By the time he was finally required to stop in 2018, At age 81, which was the upper legal age limit for donation in Australia, Harrison had donated 1,173 times. Australian Red Cross Lifeblood, the country's blood service, credited his plasma with contributing to the anti-D treatments given to an estimated 2.4 million Australian mothers and babies over the decades of his donation history. Since a single one of his donations could be processed into multiple doses, and his contributions made up a significant share of the national anti-D supply during long stretches of that period. He received Australia's Medal of the Order of Australia in 1999 for his service, and in 2005, Guinness World Records recognized him as the person who had donated the most blood plasma of anyone on record at that point, a title he held until an American donor in Michigan surpassed his total number of donations in 2022, several years after Harrison had already aged out of the program. When asked about being called a hero, Harrison would routinely wave it off, pointing out that he sat in a comfortable chair, was given a cup of coffee, and went on his way afterward, calling it no hardship at all. He continued to say, for the rest of his life, that his hope was that other people, especially those who, like him, were Rh negative and had been sensitized through pregnancy, would step forward to replace him, because the entire system depended on a donor pool that thin. James Harrison died in his sleep at a nursing home on Australia's Central Coast on February 17, 2025, at the age of 88. Tributes came in from around the world, from Lifeblood's chief executive, from medical researchers who had studied his case, and from families whose children existed specifically because his antibodies had been in the immunoglobulin shot their mothers received during pregnancy. His grandson later recounted the story of his father telling him, after his teenage surgery, that he was only alive because strangers had donated blood, and how that single sentence became the reason he gave nearly 600 liters of plasma over his lifetime. What makes Harrison's case genuinely remarkable from a genetic standpoint isn't just that he happened to be Rh negative, since roughly 1 in 6 people of European ancestry share that trait. It's the specific combination of factors that had to align in exactly the way they did. He needed to inherit the Rh negative genotype in the first place, which meant both of his parents had to carry at least one non-functional or deleted copy of the Rhd gene, a combination that, while not vanishingly rare, still only describes a minority of the population. He then needed to be exposed to Rh-positive blood early enough and repeatedly enough through those transfusions to trigger a strong and lasting antibody response, rather than a weak or short-lived one, since not every sensitized person maintains a high titer over time. And on top of that, his particular immune system needed to keep manufacturing that antibody at a therapeutically useful concentration for decades, reliably, donation after donation, which is itself a variable trait between different sensitized donors. Researchers who have studied high-titer anti-D donors as a group have found real variation in how strong and how stable individual antibody responses are, meaning Harrison's plasma wasn't just useful because he was Rh negative and sensitized. It was useful because his particular immune response happened to be unusually robust and durable, a combination of genetic background and personal immunological history that even now can't be fully predicted in advance for any single individual. This is also where the story becomes bigger than one man. Rh negative frequency itself is not evenly distributed across the human species, and the reasons why are still genuinely debated among population geneticists. In parts of Europe, Rh negative frequency sits around 15 to 17%. Among the Basque population of northern Spain and southern France, it climbs unusually high, with some estimates putting Rh negative frequency close to 25 to 30%, among the highest documented anywhere in the world, a statistical outlier that has fueled decades of speculation, much of it more folklore than established science, about the Basque population's deep ancestral isolation. In most African, East Asian, and indigenous American populations, by contrast, Rh negative frequency is typically under 5%, and in some East Asian populations it drops below 1%. Geneticists broadly agree. The modern distribution reflects a combination of genetic drift, the founder effects created when small groups of ancestral humans became geographically isolated from larger populations, and possibly some degree of selective pressure, though there is no fully settled consensus on exactly which forces did the most work over tens of thousands of years. Since Rh-negative status carries no obvious survival advantage or disadvantage on its own outside the specific context of pregnancy incompatibility, and the incompatibility problem itself is a comparatively modern medical concern rather than an ancient evolutionary pressure. What is well documented is that the practical consequence of this uneven distribution is a global shortage of exactly the kind of donor Harrison was. Countries with lower baseline Rh-negative frequency have an even smaller natural pool of potential high-titer anti-D donors to draw from than Australia does, which means the treatment that protects Rh-negative mothers worldwide has always rested on a genetically narrow and geographically uneven foundation. Medical researchers have not been content to leave that foundation as fragile as it is. For years now, teams in the UK, France, and elsewhere have been working on lab-grown alternatives, most notably monoclonal anti-D antibodies produced from cloned antibody-secreting cell lines rather than harvested from human plasma donors at all. These monoclonal versions can, in principle, be manufactured at scale without depending on the accident of any particular person's immune history. Early monoclonal candidates showed promise, but also revealed how difficult it is to fully replicate what a human immune system does naturally. Some lab-made versions cleared fetal cells from the maternal bloodstream too quickly or too aggressively, triggering unwanted inflammatory responses that a naturally sensitized human donor's polyclonal antibody mixture tends to avoid. Researchers studying the molecular structure of these antibodies have found that subtle differences in how the antibody's sugar molecules are attached, something called glycosylation, significantly affects how gently or aggressively it clears the targeted cells, and getting that balance right in a lab-grown antibody has proven harder than simply identifying the right protein sequence. In the meantime, real human donors, the James Harrisons of the world, though almost none of them will ever match his exact combination of dedication and sheer donation volume, remain the backbone of anti-D immunoglobulin production nearly everywhere on Earth. There's a reason a story about one man's plasma keeps circling back to this bigger genetic and evolutionary picture, and it's the same reason his story resonates the way it does. Every single trait discussed here, whether it's the presence or absence of the D antigen, the specific molecular route by which someone's blood ends up Rh-negative, or how strongly an individual immune system responds to a foreign antigen, is written into the genome and was inherited from parents who inherited it from their own parents, stretching back through generations of people who had no idea any of this mattered, because for most of human history it didn't. The medical relevance of Rh-negative status only became apparent in the 20th century. For the tens of thousands of years before that, it was simply a silent, invisible variant sitting quietly in the DNA of scattered families and populations around the world, doing nothing in particular, waiting for a moment in history when a 14-year-old boy having lung surgery would receive transfusions that happened to sensitize his immune system, and when medicine would develop just enough understanding of antibody biology to recognize what that sensitization made possible. That's really what makes Harrison's story land the way it does. It isn't a story about a superhuman ability or a 1 in a billion genetic mutation that gave him powers no one else has. Rh-negative blood type is common. Sensitization through transfusion, while less common, isn't extraordinarily rare either. What was extraordinary was the intersection. The right genetic background, the right early life medical event, an immune system that responded in an unusually strong and lasting way, and then 64 years of a man who kept showing up every 2 weeks, for longer than most people hold a job, despite a genuine fear of needles, because his father told him once that he was alive because of a stranger's blood. Genetics loaded the mechanism. Choice, repeated over and over for more than 6 decades, is what turned that mechanism into 2.4 million lives. It's worth sitting with how recent all of this really is. Doctors had recognized hemolytic disease of the newborn as a distinct condition since the early 20th century, but for decades, they had no idea why it happened, only that it seemed to run through certain families and certain second and third pregnancies in a pattern no one could explain. The Rh factor itself wasn't even described until the 1940s, when researchers noticed that blood from rhesus monkeys, when injected into rabbits, produced an antibody that also reacted with the red blood cells of most humans, and that some humans lacked whatever it was on their cells that triggered the reaction. It took another 2 decades of research, much of it happening simultaneously and somewhat competitively in the United States, Britain, and Australia, before teams of researchers, including physicians, running clinical trials in New York in the mid-1960s, confirmed that injecting Rh-negative mothers with anti-D antibody shortly after delivery could prevent them from ever becoming sensitized in the first place. That trial, and the ones that followed it internationally, turned a treatment that had only ever existed as a laboratory hypothesis into standard obstetric care within a matter of a few years, an unusually fast turnaround for the era. Harrison began donating blood in 1954, a full decade before anyone knew what his plasma was specifically good for. He was, in a very real sense, in the right biological position before the science had even caught up to explain why. It also helps to understand what Harrison's day-to-day donation routine actually looked like, because it wasn't glamorous, and that's part of what makes his consistency so remarkable. Every 2 weeks for 6 decades, he took time off work, traveled to a donation center, and sat through the plasmapheresis process, which separates plasma from the rest of the blood and returns the red cells and platelets back to the donor's body, a process that generally takes longer than a standard whole blood donation. He did this on his lunch breaks during his working years as a state railway clerk, and he kept doing it well into retirement. Colleagues and family members who spoke about him after his death consistently described the same thing: no complaints, no missed appointments, and a donor card that, toward the end of his eligibility, looked more like an accounting ledger than a medical record, tracking a number that eventually exceeded 1,100 separate donations from essentially one arm. All but a handful of his donations came from his right arm specifically, according to blood service records, a detail so small it borders on trivial, except that it captures just how mechanical and routine an act of extraordinary generosity can become when it's repeated often enough. None of this would matter, of course, without the underlying biology holding it together. And that's the piece worth returning to before closing the loop on this story. What sits inside a single strand of a person's DNA, in this case a small stretch of chromosome 1, where the RHD gene lives, can be functionally invisible for an entire lifetime, unless the right circumstances bring it into play. For James Harrison, those circumstances arrived early, at 14, in an operating room, through transfusions given to save his life rather than to test anything about his blood. Nobody involved in that surgery in the early 1950s could have known they were setting up one of the most consequential blood donation careers in modern medical history. That's the part of genetics that rarely makes it into casual conversations about ancestry and DNA tests. It isn't only about where your ancestors came from or what traits get passed down the family line. Sometimes it's about a single antigen, a single antibody, and a single decision made over and over again for 64 years that ends up rewriting the odds of survival for millions of people who will never know his name. If stories like this are the reason you keep coming back to this channel, the science behind the traits hiding in plain sight in your own blood, your own features, your own family history, there's more of it every week. Subscribe, and if there's a specific genetic mystery or an ancestry question you want unpacked next, leave it in the comments. We read all of them, and the ones that keep coming up are the ones that shape what we cover next.